Right. So, good afternoon. In the last class, we looked at the history of neuroscience. We looked at some key ideas on how they have emerged at various points in history. Right. We said that people had a lot of understanding of brain's anatomy at the gross scale, and only towards the beginning of the last century, people started understanding what is brain like at the microscopic level, at the level of single neuron. So that was a neuron doctrine proposed by Raymond e. Cajal. Then electrophysiology, we talked about electrophysiology and how Luigi Galvani's experiments have found that brain is also an electrical structure. It responds to currents, right? And then much more recently in the 60s, Hodgkin and Huxley have taken measurements from a squid axon and worked out the basis of electricity of a single neuron, right? Then we talked about behavioral studies and how people have started thinking about the distribution of functions on the brain, starting from a very unscientific approach like the phrenology, right? And how people have constantly argued whether the distribution of the information on the brain is local or global. So, okay, so that these kinds of history of ideas give an idea of how people have been thinking about brain. Now let us take one more step further, and instead of just looking at the parts of the brain and how what different parts do and all that, let us take one little step at a time and become familiar with the simplest data first. And since the focus of the course is on principles and not just mugging up all the details, we look at the gross facts of the brain, brain's anatomy, brain size, brain shape, things like that, and try to figure out what kind of computational meaning or significance can we extract out of this simple facts of the brain. So let us start with trying to understand the brain shape or brain structure. So if you ask why is the nervous system the way it is, right? So if you look at the nervous system, right, you have brain, which is located inside this skull, the cranial vault. It is securely located inside that bony cage. And then there is a long tail which comes out of the brain. It's called the spinal cord. And out of spinal cord and also out of the brain, a lot of these wires come out, right? And these wires fill the entire body. It's called innervation. They innervate the entire body. So the part of the brain or nervous system which is located inside the bony cage, so the brain is located inside the skull and the spinal cord is located inside the vertebral column, right? So these are it's like a fortress. Think of it as a fortress. So all the neural stuff located inside the fortress, that is called the central nervous system, right? And then you have all these wires sticking out and which fill the entire body. So all the neural stuff which is out there in the body, that is called the peripheral nervous system. Okay, so this is how central brain is structured. Now let us go deeper into it and understand, start asking very interesting questions. So one question is there any connection between brain size and intelligence? Because you will think, we all kind of generally agree that brain is the source of intelligence and, smartness and all that. So if that is the case, then people with larger brains should be smarter, right? So is there any such correlation? Because one of the first things you would like to understand about brain is what makes people smart. Because an average person is not interested, right? Which part of the brain processes vision or which part of the brain processes emotions and things like that. They would like to understand what is it that makes people smart. That's a more popular question. So people had that question for a long time, asked that question for a long time. And they applied this question to none other than Einstein to understand what is special about Einstein's brain. How come Einstein is so smart? Right. So to understand this question, after Einstein's death, they took permission from the family and probably Einstein himself, and took an opportunity to study Einstein's brain. His brain was removed after his death, and it was autopsied, and it was done by this guy called Thomas Harvey. So these guys dissected the brain, Einstein's brain, into 240 blocks, right, of about 10 centimeter cube each, and these blocks were encased in a material called celloidin, so that they can be slightly stiff. Because brain in its natural condition will be quite floppy, almost like a jelly. So in order to cut it and analyze it, it needs to have some kind of a firmness. So in order to do that, they put in this thing called celloidin. So after that, they studied these blocks extensively, did a lot of cytological analysis, that is to see what kind of cell types are there, what are their numbers, densities, right? Name it, and they have done it. Now, what have they found? Did they find the secret of Einstein's intelligence? All right, in order to understand the results of this analysis, we need to first have understand some basics of brain's anatomy, like the gross landmarks of a human brain. So you might have read in your high school biology that brain has two hemispheres, left hemisphere and right hemisphere, and each hemisphere is divided into these large regions called the lobes. These are like continents on the earth, right? These are called the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. So you can see the lobes in this picture. So the blue region is the frontal lobe, and naturally that is the front of the brain. So left in this picture is the front, and back in this picture is the the blue is the front, and the purple is the back part of the brain. So what is called back is called posterior, and front is called the anterior. So this the left-right axis in this picture is the anterior posterior axis. Then so the blue is the frontal lobe, yellow is the parietal lobe, the purple is the occipital lobe, and the green is the temporal lobe. So frontal lobe is responsible for planning, movement control, and all that. And parietal lobe has areas which process your touch sense, what is called the somatic sense, and the body image. That is, how do you have the feeling that you have a body which has so much height and so much width and so much heaviness and all those things, right? That's called the body image, and that sensation is processed inside the parietal lobe. Then occipital lobe, that is this purple region, has areas which process vision, right? And if there is damage to occipital lobe, you'll have trouble with vision. Then there is temporal lobe, that is this green region, especially on the top of the temporal lobe, where it meets the frontal lobe and the parietal lobe. There is a region which processes sounds. Okay, so obviously each of these lobes does 100 other things. They're very rich, but I just want to get the discussion going. So let us start with that. Now, if you look at the directions of the brain, so in this case, the brain is flipped here. On the right side, you have anterior, that is the front, and that's the direction in which eyes are looking. And in the left side, you have the posterior. Now, this is also referred to as rostral and caudal. So, rostrum corresponds to nose, and cauda corresponds to tail, right? So, in an animal, the front of the brain is towards the nose, and the back of the brain is towards the tail. And since humans, there is no tail, right? Also, humans stand erect, so the caudal also becomes posterior. Then the upward direction is superior, and dorsal is actually back. That is parallel to what we call our back, right? And so in an animal, the top part of the brain is aligned with the back of the animal, for a four-legged animal, for example. So therefore, superior and dorsal are often used almost interchangeably. And inferior is below, and that's also called ventral. Ventral corresponds to what we call front. That in animal, again, the lower side of the animal and the lower side of the brain, right, corresponds to what we call the front, because we stand on 2 feet. So we can introduce more terms in this context because we're going to describe Einstein's brain and what are the parts that are different from a normal brain, things like that. So understand that terminology. We need to look at a few more terms. Like I said, okay, ventral, dorsal we have seen, rostral, caudal we have seen, superior, inferior also we have seen. Then lateral means away from the middle. Lateral means to the side, so away from the center or the middle. Medial means towards the middle. Bilateral means on both sides. Ipsilateral means same side, and contralateral means opposite side. Because very often there is a tendency to, for one part of the brain to control the opposite side of the body. So therefore, we say a given part of the brain controls the contralateral side of the body. So that's why you need these terms. Or a given part of the brain sends out nerves to the same side of the body. So that's called ipsilateral. So you need these terms. I mean, biology has a lot of jargon, whether you like it or not, but I try to go easy on jargon and just use it minimally and as and when the context demands it. Then we can talk about these three sections, which define the three planes. The coronal plane is what is a vertical plane which is parallel to your front, right? And then there is horizontal plane, which is the meaning is obvious. Then there is a sagittal plane, which is also a vertical plane, but it is parallel to the middle of the brain. So since brain has two hemispheres, you can see in this picture, right? If you have to section the two hemispheres, then you have to slice it using the sagittal plane. And if you section exactly in the middle, that's called the mid-sagittal plane. So you can have other sagittal planes which are parallel to the mid-sagittal plane, but then they will be bisecting one of the hemispheres of the brain and not in the middle. Okay, so a few more terms, right? So brain is a network of neurons, and we all know that, right? You can think of it as a big graph where the nodes are either neurons or masses of neurons, big pools of neurons, and the connections or edges are made up of fibers, some kind of fibers which connect neurons. Okay, so that is generally true. But thing is, this just like Ganga is called Alkananda somewhere, and something else somewhere. So these nodes and wire go by different names in different contexts. So the nodes could be sometimes called nucleus or nuclei. This nucleus has nothing to do with the nucleus that is there inside the cell. This nucleus is a small brain structure which can have a bunch of neurons. I mean, it's a mass of neurons, or what we call gray matter. So we look at this again, when we discuss the cortex more closely, gray matter again has lots of neuron cell bodies. Okay, so that's another example of nodes, what we call nodes. Again, cortex also has lots of cell bodies, so that is nodes. Then they're also called ganglia. So ganglia are again masses of neurons, right? They are also effectively nodes. Then edges or wire is bundles of wire, so you can call them tracts. You can call them peduncles and fasciculi and nerves. So in different contexts, they go by different names. But anyway, they're all for us just connections or edges. The mass of neurons are nodes. Okay, with this little quick background, quick introduction of terminology, let us go back to Einstein's brain and see what is different about this brain compared to a normal brain. So Einstein's brain, the parietal operculum is missing. This region was missing. So operculum, what is operculum? It's a part of the inferior frontal gyrus of the frontal lobe. It borders on the lateral sulcus. Okay, let us see what that is. So in this picture, you can see. So you can see that. See this Sylvian fissure, right? And that is so this part and this little border is called the Sylvian fissure. So especially if this is the parietal lobe. This whole thing is parietal lobe. And so thing is, if you let us go back to this picture. So this is a Sylvian fissure, all right, and this is a parietal lobe. So you see that it's called a fissure because there is gap there. You can dig a scalpel there, and you will find there is a gap. Now, if you just take this part, part of the fissure, which is located between the parietal lobe and the temporal lobe, and kind of gently lift it, right, like what they're doing here with this forceps, then you will find that there is a gap there. It's like almost like opening of a cave. And if you go deep inside, then there is more brain surface there. Because brain is not like a sphere. It's not like a continuous spherical surface. It's more like think of a cauliflower if you want a good analogy, because the surface has deep undulations. Okay, so sometimes the surface goes very deep, and then you see all this. So what we are looking at is actually exactly this point. Okay, so that's what we are looking at here. So if you lift it, there is a more gap there. So this is called what is called the parietal operculum, right? And this part is missing. There's this kind of imagine that this is all very narrow and just like this and stuff like this. It's very narrow. It's like this. That is one thing. So Sylvian fissure is therefore partly absent. But this is a Sylvian fissure from here all the way here. So this is partly absent. So this cave opening is somewhat less in case of Einstein's brain. Then the inferior parietal lobe, which is this part. So this is actually Einstein's brain. The inferior parietal lobe, which is this is parietal lobe. Sorry, not so much to the back up. So this up to here. This is 15% wider than normal. So this parietal is very wide, slightly wide, not very wide. So so this is the part of the brain which is normally responsible for mathematical thought and visuospatial cognition and motor imagery and things like that. Now, okay, so but then come on, this is 15%. How is that all that makes a difference between a genius and an idiot? At 15% slightly longer, an inferior parietal lobe. No, I'm not so sure, but this is what they found. So these are the differences in terms of gross anatomical features. But there is one more thing which is in terms of the cell types found. So there's the cytology, right? So people found that in the inferior parietal area, this important location where they found some interesting differences. There are these other cells called glial cells, which are there in everybody. Glial cells, in addition to neurons, there is another class of cells which are also quite important for brain function. These are called glial cells. These cells proved to be more important only over the last 2-3 decades. Now these cells are 73% more in number in Einstein's brain, particularly in the left hemisphere, the inferior parietal area. Okay, so this is the thing. So we have seen this is Einstein's brain, and you've seen some pictures. So point is, I mean, we have a totally different image about Einstein, his intelligence, and what he's capable of, what he's accomplished, right? And we think of Einstein as some kind of a almost paragon of intelligence, especially 20th century scientific genius. So whereas when you look at the brain, it looks pretty ordinary, not very different from the brains of you and me. So what do you think of this? Okay, so thing is, we set out with a very important question, that is, what is the secret of intelligence? And can you see the causes behind that intelligence simply by examining the brain's gross anatomy? We did that. We found some differences in case of this great genius, but point is, the findings are not that dramatic. The differences are not dramatic. It's not that these are two hemispheres. He has, some 4 compartments or something like that. There's nothing very drastically different about Einstein's brain. So now what do we do? Okay, so let us go deeper and try to collect more information to go to the bottom of this question. Okay, so the initial findings with Einstein's brain are not very convincing. They don't seem to tell much about the real secret, right? So let us now look at more data. Now, first thing you would like to say is say gross weight of the brain, right? Does the weight of the brain correlate with intelligence? So let us look at some examples. Right, the newborn human, just a little baby, right? The weight is about 300 to 400 grams. An adult human, especially like a smart, IIT students, like we're just kidding, okay. So about 1300 to 1400 grams, so 1.3 kilos basically. The elephant's brain is actually much heavier. It's about 4.78 kilos. Dolphin's brain is also pretty heavy, comparable, if not slightly heavier than ours. Right, and fin whale is almost 7 kilos. Okay, so that's like a small baby. And gorilla is actually, evolutionary scale, it is pretty high, but its brain is not very heavy. It's quite small in weight. So 465. So then let's look at more animals. Rhesus monkey, right, 90 grams, very small brain. So cow is about half a kilo. Cat, very small, 30 grams. And dog, 72 grams. Rat, 2 grams, which is quite sad because rats are very smart. I mean, they can learn all these mazes. And huge research in neuroscience, huge component of neuroscience research depends on rats, and they have such a tiny brain. Viper is 0.1, right, and so on and so forth. So definitely, right off the bat, you can say that the brain size doesn't indicate intelligence, because if you look at the highest animals in the evolutionary scale, if we leave humans alone, right, the monkeys, the primates, this monkey has only like 90 grams. Gorilla is only half a kilo. Okay, and whereas elephant is not very high in evolutionary scale, it has almost 5 kilos, and a whale has 7 kilos. Okay, so these are human. They're actually much bigger than ours in terms of gross weight. So right away, obviously, that offends our ego, and we feel very bad, right, as human beings, and therefore we say, no, brain size cannot indicate intelligence. It's something else. Because it's like, so like in this cartoon where he says, this guy is saying, for a fair selection, everybody has to take the same exam. Please climb that tree. Okay, so this is actually a takeoff on our education system. But anyway, here we are doing something similar. We are comparing everybody based on, the brain weight. So simply comparing weights is not familiar because it depends. You should also consider the body weight, right? For a given body size, does the animal or that that species does it have a sufficiently large brain? Okay, then that should correlate with intelligence. That's a better parameter, right, than just looking at the gross weight of the brain. So if you take it like that, then let us look at again the data from humans. In adult human, the body weight, let us say, is 70 kilos, and the brain weight is a 1.3 to 1.4 kilos. But comparatively, if you look at the body weight of a whale, the sperm whale, it can be anywhere between 35,000 to 57,000 kilos, right? And brain weight is only 7.8 kilos. Now let us consider the brain weight by body weight. So you get pretty, depressing data because if you look at this is called E by S ratio. So E stands for encephalon, and S, that is brain, and S stands for soma, that is the body. So E by S ratio looks like this. For a small brain, it is 1 by 12. Humans is 1 by 40. Mouse is 1 by 40. So humans and mouse are on the same page. Cat is 1 by 100. Dog is 1 by 125. Frog 1 by 172. And hippopotamus is much smaller, right? And so on and so forth. So point is, this is also not very convincing. I mean, we are pretty high up, but brains are even better than us. Birds are even better than us in terms of EBS ratio. So something again seems to be wrong, not very convincing. Because see, basically, so the intention behind this whole analysis, this kind of calculation is, whatever be the quantity that you come up with, right? Which like an index of intelligence that you come up with, based on analysis of brain size and shape and things like that. That number should be highest for humans because we are the smartest. I mean, this is a kind of basic assumption, right? So therefore, if the number doesn't look like that, you keep on reworking your formulation until you find that the number that you have come up with, the index, is highest for humans. So that kind of seems to be the approach here. So bird brains have highest EBS ratio, which is not very encouraging because bird brains therefore must be accepted as smarter, and mouse is as smart as humans, right? And brain weight in vertebrate does not in general appear to increase linearly with body weight, and very smaller animals have larger EBS ratios than that of humans. And we haven't given a lot of other data, but a lot of mammals, by this calculation, EBS ratios will be higher than that of humans. So then again, we have to conclude that the EBS ratio perhaps is not a very good index of intelligence after all. Then what do we do, right? So then the thinking went like this. Let us look at EBS, the relationship between E and S, but not exactly a ratio, right? Let us assume there is some kind of a more complicated relationship. And what is that relationship? So they assume some kind of a power law. And roughly the logic behind that kind of a power law assumption goes something like this. So imagine there's a body and I have to fill this body with a bunch of nerves. I mean, I have to reach out the entire body using nerves, right? And imagine body is a sphere. I mean, no offense to anybody, just a simple kind of mathematical assumption. So therefore, the sphere is of some radius r, and assume the densities of all bodies are the same, which is also a crazy assumption, but there's a really crude calculation. Okay, so let us take then the volume as a measure of the weight, because assuming densities are pretty much the same everywhere. So therefore, the body weight is closely related to the volume of the sphere, which is 4/3 pi r cube. Now the brain size depends upon the number of neurons it has, right? Now the number of neurons, let us assume that each neuron sends out a fiber, right, to make a connection with the body. And so nerves reach out to the ends of the body. So we'll assume that the number of fibers that you have, which is proportional to the number of neurons that you have, is proportional to the body surface, because the fibers are laid to go all the way to the surface of the body and make connection. It's a very sloppy arguments, but let us see where these kinds of arguments take us. These are some fun arguments. So therefore the brain weight will be proportional to the surface area of the sphere, so that's proportional to 2 pi r square. So now brain weight E is proportional to 4 pi r square, and body weight S is proportional to the 4/3 pi r cube. Therefore, sorry, there's a mistake here. E is proportional to S to the power of 2 by 3. Okay, so this is the thing, and so that means that E is equal to some A into S power K, right, where K is about 2 by 3. So this is the kind of approach, right? Let us see where this takes us. So E is equal to some C times S to the power of R, right? R is the exponent, and E is the brain weight, S is the body weight. C, this factor is what is called the cephalization factor. Now, so the thing is, you need to fit this equation, right, to separately to different species, because you cannot fit the same curve to all species because there's so much of interspecies variation, right? So we'll fit this, this equation, this formula to different species separately. Then, so if you want to evaluate, come up with an index which says something about, right, something about the brain and some kind of brain-body relationship for that species. You take this factor, cephalization factor, for the given species, and then take the ratio of that factor C to the corresponding value for an average mammal. So C by C average mammal. Okay, and that new quantity we define an encephalization quotient. So it's a very abstract quantity of how much that species is encephalized or is endowed with a brain. Okay, so the idea here is so this is called EQ, nothing to do with IQ. Okay, kind of like IQ, but it is derived from purely anatomy. You're not looking at any function at all. So the idea here is if a species has an EQ of 2.0, it means that its brain is twice as large as expected, right, for a mammal of comparable weight and an average encephalization. Okay, so it's a fairly abstract quantity that defines, the brain and how big the brain is relative to that body weight for a given species, right? So if that is the case, then yeah. So look at this graph. In this graph, on the x-axis you have body weight in kilograms, on y-axis you have brain weight in grams, and notice that it is shown in log scale, because if you take this formula and write it in log scale, you get a linear relationship. So because log E is equal to log C plus R log S. So if you look at the log E versus log S, right, if this formula is correct, these two quantities must be related linearly. So what they're trying to show is, if you take this data from, take on the log scale and work this out for different species, right, this formula should kind of apply. And actually, interestingly, this formula applies pretty well. And in fact, look at this line is for higher vertebrates. Okay, the formula happens to be, if you just fit this, do a regression, you get E equal to some 0.7 times P power 2 by 3. So similarly here, P is S here, a slightly different notation. So for lower vertebrates, same thing is a similar law, but the factor C is much smaller. It is 0.007 times P power 2 by 3. Now note that this 2 by 3 power keeps showing up. It's somehow, so the thing is, why is that power exponent showing up like that? So we are given some loose arguments in terms of volume of the sphere and all that. Whether you take those arguments seriously or not, it's interesting that the exponent is very close to that theoretical, exponent. So now, based on this formula, let us try to, if you try to calculate EQs, people have done that. Right, we get now very satisfying results. Why is that? Because the EQ for humans is 7.4. For a dolphin, it is 5.3. And dolphin is a very smart animal. We all know that, right? And chimpanzee is close to 2.5. The monkeys, right, is again 2.09. Because all the primates are, again, there are a lot of differences among primates. This monkey, family. When it comes to intelligence, for example, chimpanzees are very smart. They can even learn language. They can even practically coin new words, and they can teach a language using some kind of sign language. People have shown that. People have trained chimpanzees to learn a sign language, and they then were able to train as baby chimpanzee and that same sign language. So they are very smart, right? So then research. So again, so what I'm saying, there's a lot of differences among the monkeys or the primates in terms of intelligence. So elephant is smaller, whale is again smaller, dog, and rat, rabbit are all very small. So EQ is more satisfying, right? And so what do we say about this? I mean, do we like this? Is it convincing? Yeah. So looks like EQ. Okay, it gives one convincing. There's one convincing aspect about it, that it is highest for humans. But on the whole, it gives a feeling that it is just fabricated so that it is highest for humans, right? Because it doesn't really tell you about how brain structure determines brain function, right? Because intelligence is all about behavior, about function, and not just about anatomy. Okay, but we were able to read some sense, read some meaning into the brain's anatomy, and these kinds of studies only seem to be able to take us only that far, right? So that concludes our first segment of this analysis of anatomy, right? So now let us take this kind of approach further, right? The approach is we want to just look at brains, anatomical features, structural features, right, and try to see what is special about them. Okay, and let's go deeper into this, and we'll see that there's a very fundamental principle when it comes to brain structure, right, which governs brain's anatomy, which can explain a lot of features of neuroanatomy. It's a very simple principle, and we'll see that in the second segment of this lecture. Because one of the questions that like I even raised in the very, I think probably the very first lecture, is that in neuroscience or neurobiology, you just keep mugging up various properties of the brain. That is, the two hemispheres and four lobes, and where is the visual area, where is the somatosensory area, and so on and so forth. But without asking, why is it like that, right? Why does the brain have two hemispheres? Why four lobes? Why is cerebellum in the back of the brain, outside? Why not inside? So think is what the logic of brain's organization, brain's anatomical organization. So that will be a more interesting question to answer rather than just collecting all the details and mugging up the details. Right. So let us kind of now work in that direction by looking at more details about brain's structural features. So let us start with the numbers of neurons or cells found in various brains. So if you look at the human brain, the number of neurons is about 100 billion. So like I said, there was a revision of this number. People find that it's slightly an overestimate. And so more recent studies say that there are about 85 billion, not 100 billion, in adult human brain, number of neurons. And if you look at octopus, which is a very smart animal, it's in fact among the vertebrates is considered like one of the smartest, right? So it has 300 million neurons. Then you have aplysia, which is a very small sea and water creature. It has only about 18,000 neurons. And C. elegans, the nematode, which is only like a millimeter or a couple of millimeters long and much thinner, right, has a very small nervous system having about 302 neurons. Then let's look at wires. So basically, if you have a graph of n nodes, right, and assuming every node is connected to every other node, right, you'll have a wire which is order of n squared, right? So I think since the brain is one of the peculiar features of the brain, his cells are very richly connected by all these wires, right? And so therefore you can easily imagine that the amount of wire present in the would be pretty large, and it is indeed pretty long, because the total length of myelinated nerve fibers in the brain happens to be about 150,000 kilometers. That's a lot of wire, because if these nerve fibers are the axons are pretty thin. Even if you take a myelinated fiber, it will be only a few microns thick, right? So the wire length is likely to be very long. Then the number of connections per neuron is about 1,000, 10,000 connections. But there's some really busy neurons like the Purkinje cells, where a single cell can have 1 or 2 lakh connections. So these are pretty rich cells. These are found in cerebellum. Then this number of synapses in the entire cortex, in the human cortex, is about 0.15 quadrillion, right? And the percentage of brain that is white matter is 60%. So by volume. So that means there is more wire in the brain than the cell bodies, which is understandable because brain is all about connections and not really the nodes. Connections are more important in the brain than the nodes. So the number of fibers in the optic nerve, just to give an idea, is about 1.2 million. Right, so optic nerve is what carries information from the retina to the brain. So that means the number of pixels, you can say, right, of information that the retina sends to the brain is about 1.2 million pixels. So which is therefore it's a very high density image, right? In fact, if you go to the photoreceptors, because information in the photoreceptors is encoded in the form of this optic nerve fiber information and carried by the nerve upstream to various brain areas. But if you look at the photoreceptors, they're even more in number. It will run into about, I think, 10 billion. Then the number of fibers in cat optic nerve is much lesser, about 100,000 fibers. Rat optic nerve is also in the same order of magnitude. Then if you take a section of the brain, in this case, it's what they are showing in the left side of the slide is the coronal section. It's a plane, vertical plane parallel to the kind of front of the body. So the section looks like this. So in the section, you can see that there are this white or creamy white areas. Okay, and then there is this, it's not gray actually, it is a slightly light brown, you can say that, right? So these light brown areas in the surface is called the gray matter and the white area is called the white matter. So basically this area is white because this is where the nerve fibers, right, go from. The brown areas or light brown areas is where you have lots of cell bodies. So here you have neuron cell bodies. And this white area is where you have wire. And particularly in this white area, you have the kind of wire that you have here, as where the nerve fibers have a coating of a substance called myelin. And this substance is white in color. Right, therefore, this part of the brain is white. Okay, so that's your so-called white matter. So the white matter, therefore, is wire. It has myelinated axons, and the gray matter is is neurons, and specifically what we call gray matter is this thin layer which covers the entire brain surface, and that is called the cortex. So, in fact, one big question I always wondered is why is there this sheet of cells all around the brain along the surface? Because I can imagine masses of neurons deep inside, like this one. So this is striatum, this one. So these are all this darkish patches are masses of neurons deep inside, okay, inside the brain. But why have this continuous certain layer of neurons running all over the surface of the brain? I never understood why it is. I couldn't even find any explanation in literature. We know that it's a fact, but why is it like that? So then if you look at volumes, when you go to larger brains, they have more white matter. There's a fraction of the white matter is higher when you go to larger brains. So for example, if you look at a hedgehog, the amount of white matter is only 13%, whereas in the human neocortex, right, it's 42%. So as the brain gets bigger, the fraction of the white matter, right, and gets bigger, because both white matter and gray matter, increase in a larger brain or higher in a larger brain, but the fraction of wire also is larger in a larger brain. That means the wire grows faster, right, through evolution. So this picture shows again the relationship between gray matter volume and the white matter volume. So the gray matter is the cell bodies and the white matter is, like I said, it grows faster, right? And the relationship is very interesting. You could almost fit a straight line quite perfectly, right, to all this. So they have taken the gray matter volume and white matter volume and in log scale and plotted all the points, and you could easily fit a very nice straight line through them. So there is a power law that seems to govern the relationship between gray matter volume and white matter volume. So this is a very interesting empirical data. So this is the kind of data. So if you again look at our favorite comparison with the history of astronomy, this curve or this straight line, for example, this graph is like one of the Kepler's laws, right? So there's raw data, which is like your star charts, right? And all these data seem to fit a certain, right, can be fitted by a certain straight line. But then we need theory which explains why is there this relationship? Why is there this wonderful scaling law that relates gray matter volume with white matter volume? I'm not sure if there is a theory which explains this, but these are the kinds of things that will be more interesting from a theoretical point of view. And that will really put the whole field on a solid scientific footing. Now, let us try to even rationalize why do the fibers grow at a faster scale? Right? Why does the wiring volume grow at a faster rate than the nodes or the neurons? So like I said, think of brain as a network, and so as a graph, right? And if you have just two nodes, right? If you have two nodes, you have only one edge. And if you have three nodes, you can have three edges. Four nodes, you have six edges. Six nodes, 15 edges, right? And this is a basic property of a graph, of a fully connected graph. These are now brain is not fully connected, right? And so in fact, a single neuron has only about 1,000, 10,000 connections. There are 100 billion neurons in the brain, but a single neuron is only connected to 1,000 to 10,000. Even though some of the very busy neurons are connected to about 100,000, but still it's quite sparsely connected, not like nothing like a fully connected graph. But still, it is right. It is so it's not exactly square. Okay, so if it is fully connected network, the number of connections will be n into n minus 1 by 2, assuming the connections have no direction. But in the brain, connections have direction because there's a connection going from neuron A to neuron B, right, is not the same as connection going from neuron B to neuron A. Connections always, information always goes in a unidirectional fashion. A given wire carries information only in one direction. You cannot go back and forth. So if that is the case, then the number of connections must be counted as n into n minus 1. Okay, so that's n, like, not this factor of half will go away. But basically, you are looking at, if you look at full connectivity, you have order n square. But for the brain, it is less than that. If you look at the power, it will be something less than 2, right? This 1.23. So it's not 1, it's not 2, it's something in between. Okay, so it's greater than 1, therefore the wiring increases more than linearly as the as fraction of the volume. Okay, that is some interesting data. Let us look at more features of the connectivity patterns in the brain. All right, you have you can say there are two kinds of connections in the brain. There are feed-forward connections and feedback connections. So in fact, if any of you are familiar with if you taken any course on deep networks or data analytics and things like that, you might have studied these deep networks, right? Because deep networks can be a very convenient tool to understand some features of the brain. Because in a deep network, you have information coming in and you have lots of layers. Information goes from layer to layer and goes to the output layer, and then you get the output. Okay, such networks where you have information going always in one direction, these are called feed forward networks. Okay, in brain also you have lots of connections which are in the feed forward direction. So this is a very similar idea. But in deep networks also, some networks have feedback connections. You can have feedback from one layer to the previous layer, right? And brain also has that. In fact, in the brain, it's quite common. It's almost the rule. This is the norm. So typically, if a brain region A in the cortex projects to or sends connections to another brain region B, this other brain region B is generally very shows very good hospitality and responds by sending connections back. There's a lot of reciprocity, right, among in the way the different brain regions connect with each other. So then again, among feed-forward connections, there are again different subcategories. So for one such category is called point-to-point connection. So I have, let us say, a brain area A, another brain area B, right? And neurons, there are lots of neurons here, and all these neurons send connections to all these neurons here. Okay, and very often brain areas are in the form of sheets or patches, right, with some area. They're very rarely like a shapeless blob, right? Very often they are like a patch of neurons. So if you look at one patch here, another patch here, and they're sending out fibers, in point-to-point connections, nearby neurons send projections to nearby neurons in A, send projections to nearby neurons in B, in such a way that if you activate some neuron in A, it will activate a unique or a neuron or a unique set of neurons in B, right? So that by looking at the response, which part of the brain of B responds, right, I can kind of make out which part of the A was stimulated in the first place. So there is some kind of one-to-one relationship between neurons in A and neurons in B. So this is called, such connections are called point-to-point connections. Then you have convergent connections where neurons in one region, right, a large region, send connections to a very small region where you have a small number of neurons. So that means a lot of these fibers converge, right, in a small region. These are called, these are also feed forward, but they are convergent connections. There are many examples of such convergent connections. For example, the cortex, right, projects to a tiny area. So this cortex is a huge area, almost surface of the brain, and large parts of the cortex project to this small region called the ventral striatum. Okay, so therefore, since a huge area projects to small area, it has to be like a many to one kind of connections, which are called convergent connections. And obviously now, if you look at the opposite direction, the opposite of it, the inverse of it, is divergent connections, where a small brain region having a small number of neurons projects to lots of different targets in the brain. So for example, this area in the brain called ventral tegmental area, right, projects to lots of the other brain areas. I mean, in fact, in humans, this tiny region very deep inside the brain projects to big part of the frontal lobe. We have seen frontal lobe, right? So it projects to big part of the frontal area. Such connections are called divergent connections. Then you also have feedback connections. And here again, there are what is called reciprocal connections, like I just said. If a brain area A, right, sends projections to a brain area B, then if B sends projections back to A, such connectivity is called reciprocal connections or reciprocal connectivity. It's also sometimes called reentrant connections. Okay, so one example of this is cerebral cortex, right? So this is cerebral cortex, let us say, that sends projections to thalamus, right? And thalamus sends feedback to the cortex. So these are called reciprocal connections. Then you have these huge loops where A projects to B, B projects to C, C projects back to A. So the thing is, there is a loop, but in this case, the loop exists, but it's immediate. A projects to B and B immediately sends feedback to A, right? But in case of loops, you have the connections come back to the origin, but after making many jumps. So another example is cortex to pontine nuclei to cerebellum to thalamus to cortex. So here I'm not expecting you to mug up all these facts. I just want to look at these patterns. All right. So like I said, from cortex to thalamus and back, that is reciprocal connections. Whereas one example of a loop is this cortex to basal ganglia. So for example, cortex, right, sends inputs to this, what is called the neostriatum. Neostriatum sends projections to what is called globus pallidus externa. This in turn projects to globus pallidus interna. From there, it projects to this place called thalamus, and then thalamus then projects back to the cortex. So you see that the loop is closed, the circuit is closed, but after many jumps, after many stages, right? And there are lots of such loops in the brain, and this is one example of such a loop. then there is a lot of information that that you can gain by simply what is called a local connectivity analysis. That is, even if you look at a small patch of the brain, let's say in the cortex, if you look at a small patch of the size of it's a square millimeter, right? Within that, you will see very rich connectivity among neurons, right? And because if you look at, we'll see this more detailed later, but if you look at a small patch of the cortex, and cortex is like a slab of neurons. Within this slab, you'll have about 6 layers of neurons, right? And then, so there's a lot of information about which layer connects to which other layer, and et cetera, et cetera. So so if you look at maybe a few hundred microns, right, size, and look at this connectivity pattern, right? And such circuits are called local circuits, right? And the connectivity among neurons in such small volumes, right? They're called local connectivity patterns. Okay, so we have seen something about brain's connectivity, right? And now let us ask a question, and with that we can stop for the day. That is, we have all these different kinds of connections. There are feed-forward connections, feedback connections, there's point-to-point, and convergent, and divergent, and long loops, etc., right? Now, what is the logic of brain connectivity? Why is it hooked up like the way it is hooked up? Right? For example, instead of this kind of very intricate and elaborate and precise connectivity patterns, because think of brain as just a big graph, right, with lots of nodes and lots of edges. Just try to just randomly shove it inside the skull. All right, what will happen if you try to shove it? It turns out that if you just randomly shove this entire graph inside the skull without taking care to place each of these nodes or each of these masses of neurons at the optimal location inside the brain, instead of doing that, if you randomly shove the whole thing inside the cranium or the skull, right, the kind of brain volume that you get will be much bigger. Specifically, if you take human cortex, right, because within the human cortex, if you take cortex as the surface of the brain, right, think of this as some kind of a side view of the brain, and this surface is called the cortex. Within this cortex, there are different parts connected to different parts, and so on, so lots of connections, right? Assume that you have this graph structure. Imagine you have these little patches of neurons. Instead of putting them in this form, the way it is in the brain, just imagine they're all separate, and you just shove them inside the cranium. Then what will happen is, if you just put them randomly, the human cortex will be 10 times larger to maintain the same degree of connectedness. So this calculation has been done, and that was what was reported in this paper by Mitchison. What that means is, the parts of the brain are packed very intelligently, very optimally inside the cranium. Okay, so to maybe they are being governed by some law. Maybe there is a meaning, there's a reason why they are packed like this, right? They're satisfying some kind of a law, right? So I don't know if you have heard about this, that when you go on air travel, especially when you make international travel, where you end up taking a lot of luggage, right? There's a very serious restriction on how much weight you can carry. So what do people do? You do something like what is shown in this picture, right? You pack everything in a suitcase and sit on it, and then bang, bang, bang. You try to pack everything in the suitcase, and that may not work, because you have to be really smart about how to pack your clothes inside a suitcase of a given volume, because volume specifications are given by your airline guys, right? So usually big clothes in the center and small clothes towards the peripheries, and so there are all sorts of rules. In fact, there are whole courses and YouTube videos that tell you about how to optimally pack, right, your suitcase with your articles, your clothes, and all that. So if packing an international luggage is such a big hard problem, imagine how much harder it is to pack a brain, right, inside a skull. Because nature has been working at this problem for millions of years. It's come up with something, an optimal solution, right? So point is, the problem of packing the brain inside the cranium is not trivial problem, right? We need to look at it more closely. So if you understand that, we'll be able to unearth a very important law that governs brain's anatomy. right? And to make progress with that law, to understand that law, we need to ask a much more fundamental question. Why do I have a brain? What if I don't have a brain? Okay, so it seems like a brainless question, but just think about it. What is the purpose of the brain? Why do animals have, or humans, why do creatures have brain? Okay, so think about that, and I want you to come up with some answers for the next class, right? And so I'm stopping the recording and